All right, I told you that there were at least, that I very quickly read over this thing and found at least 13 points in which you could argue. And on these papers you're grading, you've got to have at least 7 of these 13. You've got to have at least 7 of these 13. Okay, you ready to go? Okay, the first error is in the first column, in which he says it takes an average of 6 years between a grapevine is planted and when it bears sufficient fruit to warrant the effort of picking. And I think you could usually find a considerable argument to support the idea that the 4th year is the important one, not 6. Now, if somebody has that correct, okay, give them credit. If not, make sure on the paper that you show that you give them credit for that. That if they point out that it doesn't take 6 years between it bears, from the time it's planted to it bears sufficient fruit to warrant the effort of picking. My God, the effort of picking, you can actually make money on it the 3rd year. But he says the 6th year. So that's an error, or at least one point for discussion. Several of you come to me with a point, and so I think most of you got this pretty clear. He says to rip to 6 feet. I want to know somebody who has the equipment to rip to 6 feet. So ripping to 6 feet instead of 3 or 4 feet. 3 or 4 feet is what we normally recommend and what most of the equipment is set up to rip. Nobody that I know of. Does anybody here know of any equipment that can rip to 6 feet, or is set up to rip to 6? Yeah, but does he?
He does.
For what? I'll see. Okay, but nobody in fact, most of the coastal county soils, which this guy is talking about, you don't have soil to 6 feet.
Before we get to that one, there's one on richness of soil in the 3rd column.
All right, we'll get to it. Okay, what's your question?
In the 3rd column, 3rd page.
Okay.
Soil.
Mm-hmm.
Lines no care for soil which is too rich either.
What about it?
Well, that's one he marked as one of his 7.
Yeah.
Yeah, mine too.
Yeah. Okay, most of you did, and many of you marked that as being correct. Vines don't care for soil which is too rich either. I don't have that as a point because I sort of lean over backwards on this fertilizer business. But if most of you put that down, that's a correct statement. What do you put? He put it down as a false statement? Well, that's wrong, see. But all you got to do is get 7 out of the 13. You can put down things that are wrong. But so many of you wrote that down. I glanced through a few of these papers. Vines don't care for soil which is too rich either. And a lot of you put that down as an error. Vines don't like rich soil. That's why we can't grow very good grapes here at Davis. Well, about 1 at a time instead of 50, huh?
Okay, I believe one of your statements was that Hampshire soils are some of the best soils in the world, and that's where we grow most of the grapes.
No, that's where we grow Tokay, but we grow Tokay there not because of the soil, but because of the climate.
But they get good production out of the rich soil.
The whole point of the damn thing was that if you put it on a rich soil, you're going to get half a ton an acre, which is just patently not so. You're going to get lousy grapes, 18 tons an acre, and 20-foot cane. But
I don't see where you get that out of that stuff.
well, that little paragraph talks about laziness and
But you really don't want, but that point is not a, you can argue that point back and forth. That's why I didn't even include it as one of the points. Okay. Okay, number 3. The 3rd point is whether or not he says something about fumigation is necessary. He says, for this reason, a machine is called in which drills deep holes in the soil and fumigates a powerful insecticide, fungicide, etc. And you really don't need this unless on virgin soil, which this guy supposedly is talking about. so you don't need fumigation on virgin soils. Then another little minor point here, where he talks about this spacing of 8 by 12 foot to be ideal, and 430 vines the acre. And I told you in class that I argue with Winkler on his old book and on his new book, that not to confuse everybody with that 430 business, but it's 453 vines exactly to the acre at 8 by 12, not 430. Then he goes ahead to make a comment about you got to plant it to the contour to give the right exposure. No, he says contour is not important. Where is it? Over here. Yeah, he says, although occasionally one sees curved rows of vines, laying out such a vineyard is extremely difficult, time-consuming, and rarely offers any advantage other than aesthetics. Where is this guy? That's in the third page, opposite all those three pictures. And he says the contour planting rarely offers any advantages other than aesthetics. I don't know where he grew up, because contour planting is extremely, that's what's ruined our coastal vineyards. I used to show the class pictures of the vineyard down at Palma Sons up on the hilltop. When you look at them, you can't tell whether they're planted right side up or otherwise the other way, because so much soil is washed away that you see so many roots down below and so many arms up above. You can't tell whether they've done it this way or that way.
On the front page, they have a picture of a curve.
Yeah, but he says that's aesthetics only, which is rather a poor statement. And then the next question was one I mentioned before. He says where you plant it at the maximum exposure. And I think from your discussion on regions 1, 2, 3, 4, 5, you know that in California we're not worried about maximum exposure. In most cases we wish we had minimum exposure. So that's a bad statement. And then now the average is 6 years, rip to 6 feet, fumigation necessary. There are three important ones. I marked the ones here I thought were important. The next one coming up is important, and he says the sprinkler will protect it to 22 degrees. That's 10 degrees. And I don't believe that any of us will go on record as saying that sprinkler will protect it for 10 degrees. At 6 to 8 is probably the maximum. So that's a fairly important point. He said 90% take is okay. That's a minor point, but it's one to discuss, because we usually consider 95% as a reasonable take. The next one, which most of you caught, I hope all of you caught it, he said to put on a ½ pound of nitrogen per vine. Whew. That's only 250 pounds per acre. That's enough to blow your mind. Even a quarter pound would be too much. And it's that type of statements that can cause people who just pick up these articles and read them to lose their shirt. Who in the world, reading this article, go out and put, wouldn't know any better, you know, go out and put 250 pounds of nitrogen per acre.
He might be talking about ammonium sulfate.
Yeah, but he didn't say it. He said nitrogen. Yeah, he means ammonium sulfate, but you don't know it when you read the article. And he says a ½ pound of nitrogen per vine, which would really foul you up terribly.
I have a question getting back to the statement about the rich soil. If it's written down here, is it right or wrong?
Well,
Are we going to grade it?
you're going to pick 7. Of all these 13, I'm going to give you enough so that the person ought to get 7 of them right. You just ignore that one for the moment. Only 7 total? Yeah, I said that you've got to get at least 7. Okay.
So now, is rich soil,
What does he say about the rich soil?
he says vines like rich soil and are very productive.
How about that?
He said that the statement in there about the rich soil is false.
I guess we got through. Well, the statement in here is false. Is that what he said?
Right, yeah, he's saying
That statement is false?
is incorrect.
Okay, it's incorrect. But since there seems to be so much haggling over that one, let's throw that one out. We'll count it either way. Okay, then the next one, which is somewhere in here, I can't find it at the moment. He says mulch, that cover crop gives mulch to the soil, which is necessary or something. I've lost my copy of this, so I'm using somebody else's. Thank you. But he says something about mulch being good for the vineyard and needed, etc., which is a false statement, of course. And then somewhere later on he talks about flowering being a 3 to 7 day period. Well, it's very, very, very rare that we have flowering take place within a total period of 7 days.
What about the one where rabbits find the leaves, don't find the leaves tasty?
Rabbits don't find the leaves tasty?
After they get up above the milk carton, you don't have to worry about it because the rabbits don't like them anymore.
Well, I think that's a question that they've got other things to eat by then. But that's a point. That would be a point. Okay. If any of you've got that about they don't find them tasty on that rabbit deal, count that as one of the points. Okay. That flowering period, 3 to 7 days, though, is fairly important because flowering can go from 7 to 14 or 20 days, depending on whether we have a long, slow, cool period or a sudden week of very hot weather. Yes?
What about the statement, planting lettuce liver on its own roots is a practice due care to enhance the stage because of the wisdom?
What? I don't think anybody heard you, especially me.
What about the statement, planting Vitis vinifera on its own roots is a practice due care to chance of age because of the risk of phylloxera?
Well, see, he's talking about the coastal county, and that's a true statement.
Yeah, but well, he doesn't say necessarily. He said here, well, the guy here makes a statement from Los Angeles. Many acres in California are on their own roots. So,
Yeah, they are.
So
The majority of our vineyards are on their own roots. But he's talking about a coastal county situation.
where did he say that?
Well, Christ, can you, it says Ukiah right on the front page.
About South Coast County, Monterey.
So that I think that's an okay statement.
Another statement says, why keep planting Vitis vinifera? Vitis is
Will you shut up?
Vitis is a small letter. You got it under
Spell wrong.
It is spelled wrong? What page is this? Yeah, Vitus. I see it here now. Okay, that's a good one. Okay, count that if anybody put it down. Boy, I didn't catch that. Vitus.
What about the guy listening to his vineyard grow for 11 months?
You know, yeah, don't worry about that.
Vitis doesn't make a noise like grow.
No. Okay, the next.
I'll just about the one here that says, vineyards who do not need or cannot afford the
What?
vineyards who do not need or cannot afford the overhead sprinklers install drip irrigation.
Where's this one? And what'd you say? Now read it again.
It says,
Slowly.
vineyards who do not need or cannot afford the overhead sprinkler irrigation protection system install drip irrigation, which is especially thrifty, etc.
Well, that's a good point.
No, just do.
If you don't have enough water,
How many people are installing drip irrigation now?
well, a lot of them. A lot of them. Okay, let me give you the last two here. The last two that I wanted to conclude, which will blow your minds, are the quotations at the end, which have nothing to do with grape growing, but sort of bugged me because both these quotations were wrong. He says something about, he says, to everything there's a season and a time to every purpose. Someone said in a century past. Well, somebody said it in the Bible, for crying out loud, not in a century past. Yeah, a century past. And then the last one is what really killed me because it's my favorite quotation. Homer Khayyam says, what can winemakers buy one half so precious as stuff to sell? Which is, he got the right poem, but he got the wrong quotation, and he put it in quotation marks, which means it's a quote, which is wrong. Because it says, I wonder often what the vintner buys one half so precious as the stuff he sells. So those last two don't really count. I told you I gave you this. The 7 most important ones, once again, the average of 6 years, drip to 6 feet, fumigation necessary, which it isn't, sprinkler protection at 22 degrees, fertilizing with half a pound of nitrogen, mulching the soil, and the flowering period being so short. But any of the 13 that I gave you, if any of those people on there got those 7, plus a couple that I mentioned, it's okay. All right, Dr. Kliewer, you take over. No, you can go on all morning.
You want these exams passed in now?
I want them turned in, and I want any 7 plus those couple that I acknowledge in class to be
Okay, we're getting behind in our lectures, and we're going to have to move along. We still have the subjects to cover of flower development, nematodes, phylloxera, virus diseases, fungal and bacterial diseases, and grape insects, and we have about, I think, 7 or 8 lectures left, so we're really going to be moving here now, and if you have any questions, I suggest that keep them to a minimum and come to those 5 o'clock discussion periods on Mondays. Now, last lecture, we said that the rudimentary flower clusters are developed beginning at about flower time, and they continue for the next 2 or 3 months, and whatever flower clusters that are going to be developed have been developed by the end of August or September. And these flower clusters then are developed to the embryonic or rudimentary flower cell stage. In other words, there's no flower parts that have been fully differentiated and formed during the end of the growing season. The flower parts themselves are formed after budding out in the spring. Now, this is different than most deciduous crops, peaches and apples and so forth. At the time of bud break, bloom occurs at the same time, and the flower parts have been fully formed. So grapes are different in this respect in that the pistils and stamens, the male and female parts of the flower, are actually formed after bud break. So we say then that by late September or October, the buds are differentiated to the primordia of the individual flowers. In other words, the embryonic or the rudimentary flower cells are there, but they're not fully formed yet. And we call these clusters then rudimentary clusters, or rudimentary clusters, or fruit primordia. Now, in a cross section of a bud, as you had in your lab last year, last quarter, you remember that we said we had a primary, a secondary, and tertiary growing points, and then also a lateral bud there. And this is the leaf scar, and we said that any one of these growing points can have one or more clusters present in them. It's largely a varietal characteristic, but generally the primary growing point has a cluster or 2 on it, and occasionally the secondary and tertiary will also have clusters. However, these secondary and tertiary growing points do not always grow during the season, but the lateral growing point always breaks during the same season. Now this is a little bit hard to understand, but when the lateral bud is the buds that you've been breaking out in your lab now, and in fact the cordon is formed from a lateral bud, and these lateral buds always grow during the same season. They may grow a millimeter or they may grow several feet, but they always break during the same season. While this is not necessarily true of the secondary and tertiary growing points. So the size and the number of flowers that are going to be in a cluster have already been predetermined during the previous growing season, essentially. There may be a little bit of differentiation at the cluster tip, but essentially the total number of flowers, the shape of that cluster, and the size of that cluster has been set during that previous year. And then the individual flowering parts themselves are developed during the between 6 and 8 weeks after bud break. Actually, that you can extend that and say that from 6 to 10 weeks. There's a period, some varieties go up to 10 weeks before bloom after bud break. And we said then that well, we didn't say this, but a fruit cluster is really a fruitful tendril, and you can see this in the vineyard. I'm sure many of you out in the vineyard in your labs have seen tendrils with a few flowers on the ends of them. Well, a fruit cluster is really a fruitful tendril, and you see all stages from a tendril that's completely formed into a fully mature cluster and to those that are only 1 or 2 flowers on the end of the tendrils. So really a cluster is just a fruitful tendril. And you know, you've already been told the system in which the clusters are formed on the shoot, the discontinuous system where you have 2 clusters and a skip, and then 2 clusters or 2 tendrils and a skip and so forth. The discontinuous system. Now let's then go into this. I've written on the board here the outline that I'm going to try to follow. I don't think we're going to get through all this, but let's first talk about factors affecting development of flower clusters. And one factor that affects flower clusters is, of course, the position of the shoot on which the flower cluster develops. Now, terminal shoots generally grow first, and these generally have a little bit of an advantage in that they have a little larger leaf area. The terminal shoots generally break and grow first, and by doing this, they have usually a little bit larger leaf area, and the clusters are generally a little bit further advanced than the basal buds or basal shoots. This is pretty minor by the time of bloom time. There's usually not more than 2 to 3 days difference between basal and terminal buds by the time of bloom. Also, shoots grow earlier and faster on canes that were well mature. Those, in other words, that have large supplies of carbohydrates stored in them. Now, the position of the cluster on the shoot will also affect its development to some extent. Generally, the lower cluster is a little further advanced than a cluster above it. And this is generally true, although there are exceptions to this rule. But generally, the basal cluster is a little bigger, a little further advanced than the more terminal clusters. And this is usually what we recommend when we do cluster thinning, to take off the more terminal clusters and leave the bigger basal clusters. Although there are varieties like Cardinal, where we told you in lab, where sometimes the second and third clusters are left, because on these clusters you do not have to remove that, what we call the whittle, that tendril right at the shoulder of the cluster. Okay, and another factor that influences development of flower clusters is the number of clusters per vine. And I have some data that will illustrate this point now. This is some data of Professor Winkler. And he did some defoliation work with Muscat of Alexander and Alicante Boucher on vines, removing different amounts of leaves about 2 weeks before bloom. So this is leaves removed. And he took, well, no, I'm sorry, this is number of leaves per vine. Leaves per vine. And he had 1,800, 800, and 0 of these 2 treatments here where the leaves were removed a couple weeks before bloom. And the percent sugar in the flower was 1.6% here with no leaf removal, 1.38, and 1.0 here with all the leaves removed. So removal of leaves does greatly affect the amount of sugar in the flower or the nutrition of that flower. Now the percent, it also affects the percent germination of the pollen. The germination was 37% here with no leaves removed, 11% here with 800 leaves per vine, and 4% here with all the leaves removed. And there was similar data with Alicante brochet, about 2 weeks before bloom. So these data indicate that the more sugar, the higher the pollination, or in other words, when you have a high leaf cluster ratio, this is favorable for good nutrition of those flowers, and you get better pollen germination and better set. Now the number of leaves necessary or optimal for fruit set, Winkler did some work on this also, and he cites some figures of about 16 leaves per cluster was needed for small clustered varieties. Now these would be varieties that weigh about 1/3 of a pound or 1/3 to 1/2 a pound or less. And 16 leaves was necessary per cluster for getting maximum fruit set. For very large clustered varieties, clusters over a pound in weight, up to 40 leaves were necessary. And there's some other figures and some other work too. We have done some work in the last few years, and I gave you a couple in the lab last week. We said this 10 to 12 square centimeters per gram of fruit is needed to get maximum soluble solids in the fruit, and this worked out to be 22 to 26 leaves per pound of fruit. And this coincides pretty well with what Winkler found 30 years ago or so. Okay, another factor that affects the development of flower clusters is temperature. And I told you that temperature generally occurs 6 to 8 weeks after bud break, sometimes as much as 10 weeks after bud break. And it usually occurs when the mean temperature per day reaches about 68 degrees Fahrenheit. That's the mean, the average of the maximum and minimum. Now, blooming occurs first generally at about the middle portion of the cluster, and then it proceeds to the shoulder portions of the cluster. If this is your cluster here, blooming is usually in the middle, and then your shoulder, and then your tip portions of the cluster generally bloom last. Now, Winkler also states in his book that about 60 degrees Fahrenheit or higher is needed for bloom in pollination, and this I think he means is the maximum temperature that is reached during that day. As we've done some work in the phytotron now where we've held vines at 59 degrees Fahrenheit 24 hours a day, and you can get blooming to occur if the vines are held at that one constant temperature during the day. So I think the figure that he means in his book is that you have to have a maximum temperature of 60 degrees reached during that day in order for bloom to occur. A question?
Yeah, now you say that you can get bloom at 59 degrees, but that you usually recommend that take the mean temperature of 68.
No,
What's the 68 degree mean temperature?
well, that's when normally bloom occurs here under field conditions. In other words, if you had a mean temperature of 68, say you had a minimum of 58 and a high of 78, then the mean would be 68. That's when normally bloom occurs under field conditions. The 60 degree temperature figure that I gave you is you have to have, and that was the figure where I meant where you don't get any blooming if it's 60 degrees or below. And I think that figure is a little bit high, because we can get bloom in the controlled conditions in the greenhouse when temperatures are 58 or 59 degrees Fahrenheit. But this is much different than occurs out in the vineyard conditions, where you have a minimum down in the middle 40s and maybe a high during the 70s or 80s during the day. So it's a diurnal variation under field conditions. In the phytotron, we have this one constant temperature at 58 or 59, and you can still get blooming. That was the only point I was trying to make there. And blooming generally occurs as I think probably most of you know, over a 3 to 6 day period on a single cluster, and within a vine it generally occurs over a period of up to 10 days, maybe 7 to 10 days, a range of blooming on a vine. But within a single cluster, usually your bloom occurs over about a 3 to 6 day period. Now this low temperature also affects the pollinization and pollinization and fertilization. Dr. Winkler, I think, did some work where he cites that a period of 4 to 7 days was needed for the pollen tube to grow from the style or the stigma down to the ovule at 60 degrees Fahrenheit, a period of 4 to 7 days, while at 80 degrees Fahrenheit, a period of only 4 or 5 hours was required for that to grow. So there's a tremendous effect there of temperature on the time that the pollen, the nuclei from the pollen, to form this pollen tube to grow from the stigma down to the ovule, or ovary.
That was like at 60 degrees.
At 60 degrees, about a period of 4 to 7 days was required, and at 80 degrees, 80, 85 degrees, only 4 or 5 hours was required for that pollen tube to grow to the ovule. Okay, let's ask this question of what effect does rain have on fruit set? And there are several things that we can mention there. First of all, rain in California is almost always associated with low temperatures. When we have cloudy, rainy conditions in California, it usually almost always means low temperature. So, and we know that low temperature affects fruit set. So this is one factor of rain. Rain also would have the effect of dilution of the stigmatic fluids, these sticky substance that's on top of the stigma here. There's stigmatic fluids, and if you had a lot of rain, this would have the tendency of diluting the stigmatic fluids, and this might have an effect on fruit set, although I think this is minimal. And the reason I say that is that we've done some work where during bloom time where we run sprinklers 24 hours without any apparent effect on fruit set. So I don't think that's too important a factor, although it's mentioned in the book. Another factor is that the continual application of water can cause the pollen grains to absorb water and burst the pollen grains. So this is a third factor. And then rain can also cause what we sometimes call persistent calyptras. And this is the calyptra here is these petals that are on the outside of the flower cluster. And during high relative humidity, you don't get the drying out of the base here of this calyptra. This is where the abscission layer and these calyptras are formed, and you don't get any drying out, and these caps, as we call them, or calyptras, remained attached to the flower there and don't pop off like they normally do. So under high humid conditions, rainy conditions, you don't get these calyptras, or sometimes as they're called, caps, to pop off like they would under drier conditions. You can in fact get some fertilization and pollinization without the cap completely coming off, but it does tend to keep some of the pollen from the anther here from falling on the stigma when the cap doesn't fall off. This process of when the calyptra or the caps pop off, there's usually a burst of pollen that's ejected at that time, and it's during that time when we have pollination lighting on the stigma here, and that's when the fertilization also occurs. So if you don't get those calyptras or caps popping off, you would probably have the tendency of having less fruit set. And then a fifth factor might be that you could mention certainly is the under low light intensity that we have during rainy weather is that you have less photosynthate being produced and less carbohydrate nutrition for the flowers. And I might also mention that the varieties are extremely variable in their sensitivity to rainy conditions. A few years back there was emperors out here in our vineyard. We had some rainy period there during the bloom time, and they almost completely didn't set a single flower on these clusters, while there were other varieties nearby that set a fairly good crop. So there's quite a bit difference in varietal sensitivity to this low temperatures, to these rainy conditions. Quick question? I know, but this was an observation that we did. Most of the wine varieties, the student vineyard was right nearby, then they were setting pretty good. Now there's two schools of thought on what determines fruit set, and there's the school here that said that fruit set is determined by some hormone produced in the leaves, and this has pretty well been disproved now. There's now most of the people that are working in this field now don't consider that there is a hormone produced in the leaves that causes the fruit set. The present theory is that the amount of fruit set is determined by the supply of organic nutrients that is translocated to the ovules during this period of bloom. And this has been done by some careful work where they removed all the leaves and kept all the leaves off during the entire period of bloom, and did some other work where they could regulate the amount of carbohydrate going to the flowers, and they were in fact able to get good fruit set without any leaves at all. So a high leaf area per cluster favors good fruit set, and this is due to the favorable carbohydrates that are being produced that can then be used to produce viable pollen and also good nutrition for the ovules. Now, how long does pollen tubes stay viable in the vineyard? Figure generally cited is about 5 to 10 days that the pollen can stay viable in the vineyard. And generally, you can, if you have a 10 to 20% germination of the pollen grains, this is generally more than adequate to get a good fruit set. Remember the figures that I put on the board here when you had all the leaves on it. We said we had 37% germination here. You got the maximum germination of the pollen with all the leaves on it. We say that if you have from 10 to 20% germination of the pollen, you generally can have a good fruit set. Yeah.
I point out that percent germination of pollen doesn't mean 37% set of the flower.
This is done on auger plates.
You get about 15 to 20% of the flowers to set, and you've got a good crop.
Yes. You never get 100% of your flowers to set. In fact, it's a good thing you don't, because if you did, you'd have such a compact cluster in such a tight condition, you could have a much more favorable condition for bunch rot. So 15% of the flowers are generally fertilized. And this figure here is just percent germination of the pollen, which is done on auger petri dishes and in the laboratory. Okay, then let's go on and talk about the development of the flower, or development of the grape flowers. I think before we do that, we need to define some of these terms here. I've drawn on the board here the parts of the grape flower. This is also in your book, so if you don't have time to copy that down, you can get it out of the Winkler book. And in chapter 7, by the way, that's been assigned for your reading. If any of you want to read ahead and are ahead in your work, we're going to assign chapter 14 next, so you can jot that down in your notes, and we'll start covering that probably in the next lecture or the next couple lectures. Now, the parts of the grape flower, let's first look at the female parts of the flower, which is the pistil. And this pistil being composed of the stigma and the ovary, the stigma, the style, and the ovule, or ovary. So this whole organ here is the pistil, and this is the female part of the flower. The male part of the flower being this filament here and the anthers. So this you should have that in your notes too. These two parts are the stamen, The anther and filament are the stamen. The filament is simply the stalk that holds the pollen sac here. And this pollen sac is a bilobe thing that contains the pollen that is used to fertilize the ovules. And then we have here the ovule or ovaries. Here I've made another cross section of that, and the ovary consists of two carpels. These two carpels here, and within each carpel there's a locule, and then within each locule there's an ovary. So there's 4 ovaries. There's 2 carpels. These 2 are the carpels. Each carpel contains two locules, and within each locule there's an ovary. And if all the ovaries are fertilized, then there will be 4 seeds per berry. Now occasionally there's a 3rd carpel, and that would mean there would be 2 more ovaries, or the possibility of having up to 6 seeds per berry. But by far the most common is 2 carpels, 2 locules with the 4 ovaries, and if these are all fertilized, there'll be 4 seeds per berry. This part of the ovary is known as the gernecium, and this is the part that becomes the seed after fertilization. Now, in grapevines, there's also a pollen sac located here. It's not very conspicuous in the grapevine, and in fact, most all grape varieties have little nectar being produced in these nectar sacs. In fact, if you go out in the vineyard and you try to smell one of these flowers at the time of bloom, there's very little odor at all to these flowers. So, and the pollen nectar sacs are not very well developed in most grape varieties. Rotundifolia being an exception, though. In rotundifolia, the nectar sacs are much more developed, and there is some fragrance and odor in the rotundifolia varieties. I could also mention at this time that rotundifolias are cross-pollinated, while most vinifera varieties are mostly self-pollinated. We're going to get into that in a few minutes. Now there's three types of flowers that we can define. And these are defined by where the flower parts are attached in relation to the ovary. In the grapevine, we have the hypogynous flowers. And hypo meaning below, and this is where the flower parts are attached below the ovary. The filament and the pistil are attached below the ovary. And this was the type of flower that we have in grapes. It's also true of tomato plants also. Epigynous.
What was attached to the ovary?
The flower parts, the filament and the pistil. Now the epigynous are where the flower parts are attached above the ovary. An example of this would be apples. And then the third type is the Prerequisite. And this is where the flower parts are attached at the ovary. And examples of this is cherries, peaches, plums. This is where these flower parts are attached right here at the ovary. Now in the grapevines, I think I mentioned this, but unlike most flowers, the calyptra here, I should also mention that this corolla, there's 5 petals here that envelop the flower. If any of you want to take a look at these flowers that I brought in today, you can. You see these caps at various stages of being popped off the cluster. But anyway, these calyptra, or the corolla, there's 5 petals make up the corolla. And all these form what we call the calyptra. And in a grapevine, these do not open like they do in most flowers here at the top, but there's a incision layer formed here at the bottom. And this whole thing pops off as one cap. And we refer to this as cap fall in the grape industry. This, I think, you can see this on this slide here. Oh, this is the calyptra here. You see the 5 petals looking down. This would be looking from a top view of it. And here's what the flower looks like when that cap falls off. You can see the male parts of the vine, the filament and the anthers, and the female parts of the vine, of the flower here, the pistil. And this is what it looks like before the cap has been pushed off or pops off. This is what it looks like after the cap has come off. You can see them here if any of you haven't seen that yet. And this is what this calyptra looks like looking down on it from the top. So the corolla is made up of these 5 petals, and that whole unit is known as the calyptra. And this comes loose at the bottom here. The abscission layer is formed here at the bottom, and this is unlike most flowers. Well, I think we better stop there.